This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 378, recorded on February 26th, 2016. Hi, everybody. I'm Vincent Racaniello, and you are listening to the podcast all about viruses. Joining me today from Western Massachusetts, Alan Dove.
Good to be here.
Have you got warm or cold weather?
It's kind of blustery. It's really windy. It's only down to 34. It's above freezing.
Yeah, same here.
Fahrenheit, but just really, really gusty winds.
Did you have a lot of rain this week?
Poured.
Yeah, same thing here. And now it's sunny, clear skies.
Yeah, it's supposed to be nice for the weekend.
Very nice. Odd weather, though, because earlier in the week it was warm, right?
Right. We had brutal cold and then warm and then pouring rain and now blustery. And yes.
Also joining us from southeastern Michigan, Kathy Spindler.
Hi, everybody. It's great here. It's bright and sunny, and it's 31 degrees Fahrenheit, feels like 24, and that's around -1 Celsius. So we got a ton of snow. The rain that you guys got came for us as wet, heavy snow. I was worried about breaking my wovel, but it survived.
How many inches of snow did you get?
Oh,
6?
I'm holding up my hands now. It's 6 or 8, because it came in 2 chunks. And yeah.
That could be the last of the season, right?
Yeah, we had a big snowstorm one March 15th, and someone told me this yesterday that her child was born in April, and it snowed that day. Yeah, so.
I remember it snowing in April once here, but that's pretty rare.
Yeah.
All right, we have 2 guests today, one here and one in Chicago. And our Chicago guest is at Northwestern University Feinberg School of Medicine. He is in the Department of Microbiology and Immunology, which is our namesake here. And this is his second time on TWiV. And I'm going to let you guys guess because I've given you enough information to figure out who it is. So write down your guesses. It's Greg Smith. Welcome back, Greg.
Thanks, Vincent. There's a couple other virologists in the department, though, I think are fair guesses as well.
Well, that's right. So on that TWiV, it was you, Bob Lamb, and was a student.
Andrew Caraba from Rich Longnecker's lab.
Yeah, and who else was on the panel?
Sarah Connolly, who is a postdoc in the same lab.
All right. So, but Lamb is out in the Evanston campus.
That's true. Right? So you actually, that's a good clue. So if people are smart enough to know there are 2 campuses and who's where, they should be able to get the answer.
So if we do live streaming, we could have a real contest, and people could guess. I could get a winner, and I could send them something.
Right.
Like Greg Smith's autograph.
A highly valued commodity, sure.
We also have an in-studio guest who is not Dickson Despommier, because he's not a guest. He's a part of the show. Dickson will be along later, as usual. And this is an old friend of the show, Mark Fuccio. Welcome.
Hello, Vincent. It's good to be here.
An old listener, and in the old days of the show, a supporter through Drobo. Many of you may remember the Drobo contest, and some of you have Drobos if you're still listening. So we thank you for that, Mark.
Those were good old days. Things were a lot smaller and simpler then.
It's true, and maybe TWiV was a little shorter then.
It was.
As well.
Maybe.
Anyway, you're in New York City on business, and Mark always asks whether he can stop by, and this time it worked out, and you're always welcome, Mark, to come by. I hope you enjoy it. And by the way, Mark, we're going to talk about viruses today.
I imagine that. What else would you do on This Week in Virology?
Right. Well, we could talk about stump grinding.
Yes, front-end loaders.
Weather, front-end loaders, lots of other things. Then people get mad at us. But we're going to talk about viruses. And we have Greg here to talk about that. And this episode of TWiV is sponsored by the 32nd Clinical Virology Symposium. Abstract submissions now open for this meeting, which will take place in Daytona Beach, Florida, May 19th through the 22nd. And the deadline for submission is March 17th. What you need to do is go to the website, asm.org/CVS2016. You can register for the meeting, and you could send in your abstract to talk about your work, what kind of stuff are they going to talk about at this meeting? Rapid viral diagnosis, the clinical course of viral infections, and how to prevent and cure or treat viral infections. The audience is over 1,000 people, a mix of researchers and primary care physicians. So do check it out. It's not just 30 seconds long. It is the 32nd in a series of symposia. Last year was the 31st. Next year will be the 33rd. Check it out at asm.org/CVS2016. And we thank ASM for their support of TWiV. Now, Greg Smith, we did do your history way back when. What episode was that, Kathy? Do you know?
I haven't looked it up yet.
Oh, I thought you would have.
I was thinking of it, and then I got distracted.
It was back in 2012.
2012? Let's go through your history briefly to remember. Where are you from originally?
So actually, I was born just outside of Chicago, ironically. I say that because my family moved to Southern California when I was about a year old. And I went to college at University of California, Santa Barbara, microbiology. And then I went out east to the University of Pennsylvania for graduate school, and then Princeton for my postdoctoral studies with Lynn Enquist.
Your PhD was at Penn State, you said?
No, I'm sorry, University of Pennsylvania.
UPenn in Philadelphia?
Philadelphia, yeah.
Who did you work with?
Guy's not there anymore. Dan Portnoy. I don't know if you know him.
Oh, I know Dan Portnoy. He's out in California now, right?
He is in California now. Yeah. So we were doing bacterial pathogenesis, and I was, and specifically a bug called Listeria monocytogenes.
And that's why your Skype handle has Listeria in it.
There you go. Now you connected all the dots.
So Dan Portnoy used to be here in New York City.
Back when he was a postdoc.
Yeah.
Yeah.
So I was at a meeting with him, and I roomed with him. It was in Switzerland, actually.
I'm sure that's fond memories.
And so we came back after the meeting. It was like midnight or whatever. And we're lying there in bed, and he just starts talking. Okay, so I listen and chat, and then, okay, I figure he's done. And then he starts again, and he went on for an hour just talking. You know he likes to talk a lot, right?
Dan is a character.
So that's your complaint about Portnoy?
I'm not. Oh!
Oh, boy.
That's right. That's my complaint about. You probably heard that a lot, Greg, right?
Well, I'm sure. I know Dan has. I've kind of heard it from the side.
Yeah. And, of course, in Princeton, you were a postdoc with Lynn Enquist.
That's right.
And I remember you from those days. I don't know. Do you remember me visiting to teach at all? It was a long time ago.
Oh, gosh, I don't recall you coming to. I imagine you must have been doing polio then, huh?
So you know that course he teaches, the virology course, right?
Yeah, he's gotten some nice awards for that.
Were you a TA for that course?
I was 1 year.
Yeah. I seem to remember you, but it's a long. What are we talking about? The 80s or early 90s? No, it can't be the 80s. You're not that old, right?
No, this would be mid to late 90s.
To late 90s. Okay, yeah. Anyway, a good time there, and now you've been at Northwestern for how long?
Well, I started at the tail end of 2001, so that's a little over 14 years.
So you are tenured, full professor, extremely well funded, huge lab, right? You've got it all made.
Gosh, wouldn't that be wonderful?
I don't mean for you to answer any of that. I'm just joking.
No, it's none of it. Well, I do my best, but I should say I'm an associate professor, not full.
But you're tenured, though, right?
I'm a tenured associate professor. That's right.
All right. Well,
he was on TWiV 185.
Thank you, Kathy.
Which has a great episode photo showing showing Greg in a I guess I would call this a plaque assay bunker.
Oh,
that's not me.
That's the student.
Yeah. Oh, that's a student. Oh, okay.
What was his name again, Greg?
That's Andrew Carabra.
So Andrew was the guy who got me the idea of building a wall of polio, because he sent me this picture of him with a bunch of plates around him. It was a few feet tall. It wasn't huge. It was in his little cubbyhole or cubicle area, so he couldn't make it so big. And then when I came back, I said to my postdoc, I said, you should make a wall out of those plates, and that's where it started. And now it's behind Mark here in the TWiV studio.
I remember seeing a picture of, I think it was with Blossom Demagno. Was that there?
Yeah, that was. She visited a little while ago.
Gotcha.
Okay, and that was a very good episode as well. I remember I had Bob Lamb on that, and this is the last story I'll tell. I had Bob Lamb on that episode because he's always such a talker. And I introduced him, and he said, nothing. And he said, well, you put a microphone on me, and it's really inhibiting. I would never have believed it. So, Greg, if you ever want to silence Bob.
Bring a mic, huh?
Just clip a mic to his lapel, and apparently that automatically does it. Bob is full of stories and gossip, and I wanted him to tell some of those, but he didn't. All right, so the reason Greg is here today is last week we had a lovely conversation about how viruses invade the central and peripheral nervous systems. Mm-hmm. And I was made aware of his paper, which came out at the end of last year, I think, October 2015 in PNAS. And I looked at it while he was talking to me, and I said, my gosh, this is perfect TWiV material. So here we are. And the paper is Dynamic Ubiquitination Drives Herpes Virus Neuroinvasion. And I'm really glad you're here, Greg, because after I read it, I had so many questions that I feel it would have been hard for us to discuss it well. So I'm glad you're able to tell us all about it.
My pleasure to be here.
So can you give us the backstory, sort of what led to this paper?
Well, so, backstory could start in two ways. I guess the big backstory is just understanding a little bit about herpes viruses and what they do in order to survive in nature. And many of them are neuroinvasive. And so along that theme, there was a discovery that was made by Hida Plu some time ago now, where he was looking for viruses mostly that express enzymes called deubiquitinases, or DUBs for short. So these are proteases that cleave ubiquitin off of proteins that first have had ubiquitin put on them by an E3 ligase enzyme. And he basically found that in some lysates infected by various viruses, you could detect a deubiquinase activity that wasn't present in the uninfected cells. And the one that stood out to him more than any other was in cells infected with herpes simplex virus type 1. Mm. And so he discovered that there was this deubiquinase activity that presumably was virally associated. And to make a long story short, that proved to be true. He identified it. It was a novel type of sequence. So it was never picked out before from just sequence gazing, and he was able to get crystal structure and do some mutagenesis work with some colleagues. And it didn't really amount to much. People made catalytic inactive versions of this enzyme in the virus and didn't get very profound phenotypes in culture. But then back in 2009, I had this wonderful student who was actually a neuroscience student in the lab, and she wanted to pursue this some more and made a catalytic mutation in a virus called pseudo-rabies virus, which our lab loves to study, and it's Lynn Enquist's favorite virus. Mm. And that virus is extremely not only neuroinvasive but neurovirulent, and so it's
also a herpes virus.
Yeah, I'm sorry. That's a very important point. Yeah, yes. Thanks, Kathy. It is a herpes virus, despite its slightly confusing name. It has nothing to do with rabies, except
that it's pseudo-rabies.
Pseudo-rabies because it presents like rabies in a dog that's infected. They look like a rabid animal, but if you get bit by it, you don't contract any disease. So it's pseudo-rabies. And but it is in fact, just like Kathy was pointing out, it's a herpes virus. And so that virus then is very virulent. And so if you put it into a rodent, mouse or a rat in a laboratory setting, the animal will succumb to the infection typically in 2 days.
So if you put it in peripherally, it gets into the brain, yeah, and it causes a very severe, quickly progressing encephalitis. And so it turns out if you mutate that catalytic site in that deubiquinase enzyme of the virus no longer goes to the brain after peripheral inoculation. And so we did a lot of work to try to track down exactly why that was, and we were able to describe it in quite detail, but we didn't have any understanding of the mechanism at that time. Let me ask you, so that the deubiquitinase is part of a viral structural protein that is packaged in the virus particle, right?
That's
called PUL36.
It's called PUL36, P for protein, UL for unique long region gene, and then 36 is the 36th gene from the left, essentially.
So I noticed in the introduction you spelled that out, which herpes virologists never do.
Right.
Did the reviewer or the journal make you do that?
I can't recall if that was the case. It might have been that we were asked for it, but Lynn's, I have to, praise Lynn as a wonderful advisor. He besides teaching me how to be a good researcher, he also is very good at instructing me how to become a good communicator as well.
Mm.
So now PUL36 is this relatively large protein, and the deubiquitinase, the DUB, is just a little piece of it, right?
Yeah, it amounts to about 8 to 10% of the total mass of this protein, and it is an extremely large protein, which is it's somewhat unusual for, in virology. I mean, herpes viruses are fairly large, complex viruses, both structurally and genetically to begin with. But even larger viruses, such as the pox viruses that like Rich Condit studies, those viruses, even though they're bigger, they don't encode single proteins of that size like this one. And so there's something weird about this, but the coding sequence for this protein is is a little bit bigger than polioviruses, Vincent, which is your favorite. So it's a very large, complex protein for a virus.
So this PUL36 is between the envelope and the capsid in the virus particle, right? And when the virus, when the capsid is liberated into the cell, there's membrane fusion with the virus. Where does the DUB go? I keep calling it DUB, and I'm going to end up doing that. Where does the PUL36 go?
Well, I think either's fine. So the PUL36, so the area that you described there in herpes virology, we call that area the tegament, which is, I think other people that study envelope viruses would call it matrix. It's basically the same idea, but the tegament, it's just more proteins there, it's more amorphous, and all of that material upon when virus makes cell contact and there's the fusion event, all that material gets deposited into the cytosol of the cell. And so the tegament's particularly interesting, I think, in the sense that if a virus wants to start changing the physiology of the host cell, this is an opportunity to do it in many different ways before the time that's necessary to get viral gene expression kicked up and going. So it can start changing things quite quickly. And so the DUB falls into that category. It's being put into the cytosol and immediately can start acting on substrates in that cell. In this particular case, it's bound to the capsid surface of the particle. Okay.
So you told us what pseudo-rabies virus does when you infect a mouse. It gets into the central nervous system, right? Now, some of the experiments done here are... correct me if I'm wrong, there with herpes simplex as well, or is it all pseudo-rabies? I don't remember.
Experimentally, it's all pseudo-rabies, but we'd have some comparison with HSV based on sequences and some mass spec work.
So for HSV, just give us, if you inoculate a human or a human gets inoculated early in life, what happens to the virus?
Depends on how early and also if the person is in good immune standing. In a normal circumstance, let's say you're 12 years old and your grandmother comes up and she has got a cold sore and she gives you a nice big kiss on the cheek, then you basically now have virus that has the potential to be replicating in the epidermis, particularly mucosal epithelia. And then the virus has evolved to very effectively spread from those sites of infection deeper into the tissue, to nerve endings. And that will then bring it to the peripheral nervous system, such as sensory ganglia.
Right.
It's where pretty much resides.
Right. So it penetrates peripheral nerves. It gets in the nerves, travels to the nerve body, the ganglia, and then stays there for the rest of your life, basically. Right?
For the rest of your life. And that's why in our age group, its estimates are that 80% or more of us are carrying this virus and are infected.
Yeah. And then periodically it gets reactivated and you get a cold sore and you spread it to someone else, right?
Or you don't get the cold sore and you spread it to somebody else. It doesn't always manifest as disease.
Yeah.
But that's the classic latency that you're referring to that's known for, that these viruses are known for.
Are the pseudo-rabies viruses known for latency also?
In their native host, so pseudo-rabies' native host is actually pig. And so in pigs, it does in fact establish lifelong latency.
And the pigs pass it to each other.
They do.
They kiss each other and they pass it among them.
Yeah, and it's more virulent than in pigs than simplex is in humans, and so particularly for the young pigs, it can be quite lethal.
Okay, all right. So now tell us what you did in this paper.
Well, so following up on that DUBiquitinase, that DUB activity, we had some preliminary reasons to believe that what it was acting on might be itself, and so. When you have an enzymatic activity like that, the first thing you want to know is what are the substrates. And broadly speaking, you could say, well, perhaps the substrate is a cellular protein that it's acting upon, or maybe it's acting on one of its own proteins, one of the viral proteins. And certainly, or both could be true as well. And so we had some preliminary evidence to indicate that it could be acting on itself, on the actual PUL36 protein, this large protein. And so we decided to pursue that by kind of a very simple-minded approach, which was that the ubiquitin that when it gets put onto a protein, it's conjugated onto a lysine. And so you could just go and mutate lysines in this protein. And so I had at the time, young, naive student who, when I told him, let's mutate some lysines, it's like he was all for it. Of course, the next question is, this is a big protein. How many lysines are there? And the answer was there's over 50. And so that was going to be more than a graduate career of nonsense if you were to do that. And so we basically just said, let's try to intellectually focus this down a little bit. And what we decided to do was just look at the published sequences of this protein, or the predicted amino acid sequences from there are various different neuroinvasive herpes viruses, some of man, some of different veterinary animals. And we looked at those, aligned them, and we saw that of all these different lysines throughout the protein, there's only 4 that were invariant in the neuroinvasive herpes viruses. And so using that, we decided let's just make 4 mutant viruses, one mutated at each position, so that we would change the lysine to an arginine. And as I'm sure, as your listeners know, that conserves the positive charge on that amino acid, but makes it no longer eligible for ubiquitin addition at that position.
Right. And ubiquitin is a kind of a general purpose tag that does a bunch of different things in the cell, right?
That's right. It's a small protein, and I think of it as just a post-translational modification. So in that sense, it can be very much like phosphorylation. Obviously, the molecular weight of ubiquitin is much bigger than a phosphate group, but the biology of it can be quite similar.
Right. So this is used to target things to different places within a cell in many cases.
Right. And many people, when they hear ubiquitin, the first thing they think of is a polyubiquitin chain that can form on a lysine of a protein that will target it for degradation.
Right.
And although that's the most common thing that you hear discussed, that's certainly not the limits of the biology here. Different modifications can cause proteins just to change conformation or go to different parts of the cell without invoking their degradation.
It's like a baggage tag.
Yeah.
Yeah, and it gets ripped off sometimes.
Yes.
Well, I do live right next to Chicago O'Hare.
Gets ripped off a lot, yeah. So you did these 4 lysine changes in the pseudorabies virus, right? Not herpes simplex.
Not herpes simplex.
Okay. And you had 4 different viruses. That's it. So that's a good project, right?
I think that was a good starting point for a project. That's right.
All right. What were those like? Those 4 viruses?
Well, so this was, I think, stunning observation number 1 that told us that the project was going to have some legs, and that was that it could have been nothing. I mean, these are very subtle mutations that he was introducing, but it turned out that when he first starting off with simple virology, these viruses are cytopathic, and they make very nice defined plaques in a monolayer. So you can do a nice plaque assay with herpes simplex and pseudorabies virus. And when he looked at those plaques, he noticed that for 2 of the 4 mutations, they produced plaques that were slightly smaller than normal. And that in itself was not a stunning observation. But then working with some colleagues at the University of Nebraska, Patty Solers and Gary Pickard, who are longstanding collaborators who are neuroscientists, they went ahead and just did a quick experiment for us to save us some time, and they put the virus into the nose of some mice. And so this is just a way to quickly look at the virulence property of the virus. Like I said, small animals like that succumb usually in 2 days to these viruses. And sure enough, that's what you see with the wild-type viruses, and it was also what we saw with 2 of the mutant viruses. But 2 of the others showed gross attenuation in their virulence. One was absolutely avirulent. And that one, not only did the animals not succumb to an infection, but they didn't present any symptoms of a disease. They just continued to eat and move around and gain weight like a happy mouse would. The other one showed some lethality, but with a very delayed kinetic. So instead of 2 days, it was, I think it was like over a week, as I recall, or right about there. And then they would start succumbing slightly to a disease. Not all of them did, so it was more sporadic. And so point being is that those 2 lysines seem to be very important for the virus's virulence in this animal model.
Did these viruses that were avirulent or had reduced virulent, were they replicating in the mice?
At that point, we didn't know. Yeah, but they were replicating, as it turns out, quite well in culture. Even though they had a small difference in plaque size, if you do single-step growth, they were wild type.
Okay. So assuming that these residues are ubiquitinated, which you don't know at this point, right?
That's right.
The hypothesis would be that ubiquitination seems to be important for neuronal infection, right? Is that fair?
It's fair. It was at this point still a bit of a stretch, and I would never have said that at that point. But that's certainly where things started going.
Yeah, on TWiV you could say that. We're not peer reviewed, right?
Right.
Or maybe we are actually. We get lots of letters. Smith can't say that.
Well, you'll have to let me know what letters we get in that regard.
Yeah. Okay, go ahead.
So, right. So once we had that really appealing piece of data, we decided this was worth the time to look a little deeper. And one of the things that my lab has been doing a lot in the past was to understand the neuroinvasive process. We often will model it using primary neurons in culture. Mm-hmm. And we typically use sensory neurons for this purpose because that's the type of neuron that the viruses normally establish their lifelong latency in. And what's nice about this is you can make these viruses even though they're too small to see, even with a standard microscope typically, you can make them fluorescent by incorporating in GFP or a related protein and look at structural components of the virus inside cells by live cell imaging using fluorescence microscopy. And one of the things that we do a lot, and I pioneered with Lynn Enquist when I was a postdoc with him, was the ability to watch these viruses moving in the axons or the nerve fibers of individual sensory neurons in the culture dish. And it's an amazing thing to see. There's plenty of videos online that people can go to where you can watch these individual viral particles zipping down like little bullets down these nerve fibers and heading towards the cell body of the neuron and ultimately the nucleus.
These are wonderful movies. Absolutely. Yes.
And so if you look at the mutants' ability to do this, things got a little interesting. So one of the mutants, as it turned out, had some difficulty. And the other mutant, the one that was severely attenuated but had some virulence left, was wild type in this regard. So right away we saw that these two different mutant viruses that were both attenuated were attenuated probably for different reasons.
So what virus are we talking about here? Is it simplex?
No, it's still
PRV?
It's going to be PRV except for some mass spec work.
And this is in these neuronal cultures where you add virus, and then you can see the virus getting into the axon basically and moving towards the cell body, right?
Moving to the cell body inside of the axon, so it's intracellular.
Okay.
Yeah.
So in both cases, so wild type obviously gets in and is transported, and again the two mutant viruses, they get in to the axon.
They do get into the axon. Yes.
They have problems moving around.
Well, one of them was okay. And so for that one, we didn't know precisely yet why it was avirulent in the animal, but the one that was completely avirulent in animals, it turned out it was transporting down the axon with an aberrant kinetic associated with it. So basically, it's kind of interesting when one of these viruses is moving in an axon, it's mostly moving towards the cell body, which will get it to the nucleus of that neuron, which makes sense scientifically that that's where it's going to deposit its DNA and establish a latent infection. Right. But when you watch it very closely under the microscope, if you get if you get good resolution, you can see that the motion isn't continuous. It kind of pauses and goes, but some of those pauses you can actually see it's backing up and going what I would refer to colloquially as the wrong direction. Right?
Do they always go to the cell body? Do any of them stay in place near the axon?
So they always the preference is that they go to the cell body and to nuclear pore complexes at the nuclear membrane, but it's a really good question, and some of them it doesn't seem very often that they get stuck along the way, but we have some, if there's time later we can talk about it. There's some mutant phenotypes that match that kind of description.
So if they get stuck, let me know if this is going off course. If they get stuck, does it have any other manifestations in terms of symptoms?
No. So if it gets stuck, it's not going to propagate any further. And so it's got to make it to the nucleus so that it can start replicating and then causing any kind of disease manifestation. Except to say, if you go back to the actual infection in an animal, it's going to be replicating out in the peripheral cells like mucosal epithelia before any of this happens. And so disease can be associated with replication at the periphery that does not involve the nervous system, but different kinds of disease. But if you're going to get a neuroinvasive disease, it's got to be able to make it to the nucleus.
Mm-hmm. All right, so now we know that if you prevent ubiquitination at 2 lysines, you have issues moving in an axon, right?
Well, at one lysine, you have issues moving in the axon, and the other one is avirulent for some other mysterious reason.
Because it can move to the nucleus, right?
It can move to the nucleus.
Got it. Okay.
All right. So the latter one that can move to the nucleus for this study got shelved at that point, and so we started focusing on the particular lysine that was made the virus completely avirulent in the mouse model, and also showed that it had aberrant transport in the axons. And you can actually, and it's a difficulty with just an audio podcast I can't show you, but there's associated with this, there's this paper, there's a supplemental movie where you can actually look at the motion of the wild type virus side by side with the motion of this mutant, and the aberrancy, I think, by eye stands out quite well. But of course, we took some time to quantitate that motion and demonstrated basically what we were seeing is that the viruses were stopping much more frequently than they normally would, and they were also going in that wrong direction. They're backing up much more frequently than they normally would. And the interesting thing here is that because of all of this, you have a virus now that normally is like I said, it's like a marathon runner. It's like a bullet. It's going down that axon towards the soma. It's a virus on a mission, and it just keeps on going. But if you make this mutation, that marathon runner is now like a marathon runner that's had too much to drink, and now it's kind of meandering and going back and forth and forgetting what it's supposed to be doing. And like I said, the phenotype, you can see it by eye, and you can quantitate it quite well just by looking at that motion. And so that drunk marathon runner was the phenotype that we had, and at this point, of course, we still didn't know exactly why, but it still went in the right direction most of the time. So we refer to that as net displacement. If you look at where the particle is in the axon at any given moment, let some determined amount of time go by, let's say 10 seconds, and ask where does it end up in that time frame. With a wild-type virus, it's moved much closer to the neuron's nucleus. But with this one, it's still doing that, just not as well. So the distance is decreased, but it's still going, on average, the right direction. And yet, it doesn't ever make it.
That's why it's not virulent, presumably, right?
That's presumably why it's not virulent.
Yeah.
And why it doesn't make it with a reduced kinetics is something that we haven't figured out yet, but I think it's quite interesting.
Slow and steady does not win the race.
That's right.
Or slow and unsteady, anyway.
So, I think somebody once said that with herpes viruses, you can't run, but you can hide. I would say you have to run, and you also have to hide. I think that's the success story here with these viruses.
That's great. Okay, so what's the next step then?
So the next thing that we did, we confirmed that the ability of the viruses to make it all the way to the nucleus or not after the retrograde transport event, and then we did some mass spec work. So then we went back and asked the question: okay, we have some wonderful phenotypes associated with the mutations, but does this have anything at all to do with deubiquitination and ubiquitination? Getting back to that previous question. And so, as you pointed out, Vincent, the hypothesis you could say is that these lysines must be getting ubiquitinated during infection. So is that really true? That was the next question. And to approach that, we took a mass spec approach, and it's an approach called UBscan that a company named Cell Signaling developed. And basically the idea is that you can just do a triptych digest of proteins like you would for most mass spec approaches and look at those peptides. If a protein has a ubiquitin conjugated onto its lysine, when you digest with trypsin, not only are you digesting up your protein of interest, but you're digesting up that ubiquitin that's on there too. But one of the cleavage sites for trypsin in the ubiquitin peptide is right near its C terminus. It's just 2 amino acids in from the C terminus. And so when it cleaves, it leaves behind the last 2 amino acids still conjugated to the lysine of what now is a peptide derived from your protein of interest. And so those two glycines, the company had made a nice antibody specifically to be able to enrich for those diglycine motifs that are left behind after chewing up your protein. And so you can then enrich for those peptides that presumably had the ubiquitin on it during the infection and say, well, where is it? And so what we did is it was a comparative analysis where we looked at the wild-type virus during infection, and we looked at the deubiquitinase mutant. So this has had a simple cysteine to alanine point mutation that inactivated that deubiquitinase activity in the same protein, and asked, could we see differences? And we did this study both with PRV and herpes simplex virus type 1. So the answer was we found lots of things. Tons of cellular proteins came up in the screen that are going to be subject for future studies. But what was particularly nice is that when we looked at all the viral proteins, the viral protein that showed the most modification when the deubiquitinase, I'm going to try to take this slow, when the deubiquitinase is mutated, it had more modification, it was hypermodified compared to when it was not when it was wild type. So the idea is if you get rid of the deubiquitinase, then you're going to allow ubiquitin to accumulate and stay on the target sites.
And that's done by a mutation in the D-ubiquitinase.
That's right. That's a C to A, a cysteine to alanine mutation, which kills its enzymatic activity.
Okay. And then when that enzyme is dead, that same protein has more ubiquitin on it.
Because it can't take it off anymore.
Yeah.
That's right.
Right.
And so you look for that enrichment. And so the protein, of all the proteins in both of these viruses, herpes simplex virus type 1 and PRV, the one protein that showed the most hyperubiquitination when the DUB was mutated in the respective viruses turned out to be this protein, PU36. So that was the first nice thing. And then when you mine the data a little bit more and look at specifically what peptides from this protein are the ones that have these modifications, it turned out that in pseudorabies virus, it was the position that just lit up like a Christmas tree. It was that one position that we had been mutating, that we've been discussing all along, the one that makes it avirulent in animals and gives it this transport drunken marathon runner phenotype.
It's 442, right?
Yeah, we didn't actually say the numbers, but that's the position.
Yeah.
Right.
Yep.
So you did not have to do the experiment where you did this whole thing with the mutant virus because, in fact, you could see that it was ubiquitinated in this analysis, right, with the DUB mutant.
Right. So it's a little, obviously by working with a mutant to see this, it's a little bit of an artificial scenario.
Yeah.
And I should also say that in order to get enough material, we didn't do this in neurons. We were doing this just in transformed epithelial cells. But nevertheless, the results were consistent with the biological phenotypes that we were seeing.
All right. So now we know that K442 is ubiquitinated and that some of the phenotypes you've been seeing are most likely due to interfering with ubiquitination, right?
That's right. And not only is it ubiquitinated according to this assay, but it also says that the DUB is autocatalytic at that same site.
Right.
So we learned both of those things.
All right. So now you did an in vivo experiment, right, in a rat.
Yeah, in a rat.
Tell us about that.
Well, so the reason as most people know, when working with a small animal model, people tend to use mice for a number of reasons. Some of them are very just practical. Rats, though, have a long-standing history in the neuroscience field. So there's, as far as when it comes to mammalian neuroanatomy, the Paxonos textbook is the classic book on this, and it's all about the rat nervous system. And so my neuroscience colleagues love to use the rat for that reason. It also provides a little bit easier use if, in terms of when you're doing some inoculations in the specific tissues, everything's bigger than a mouse, and so it makes it a little bit easier to do the viral deliveries. Yeah. And so when you do a viral delivery in these systems, and I should say that a lot of this work was being done by Lynn Enquist long before I ever got into the field. So he really pioneered these kinds of models with his colleague Pat Card at the time. But so when you do these experiments, one of the nice thing to do, and even though it sounds a little unusual, is you can put the virus into the eye. So you can literally stick a needle in its eye and let the virus disseminate from an internal location within the eye.
So you're going right into the aqueous humor, right?
Yeah. So that's the fluid right behind your cornea. That's
vitreous humor, right?
The vitreous humor. That's right. Yeah. And so you can put it by putting it in there. You get it right between the lens and the cornea, and there's some nice epithelial tissues in there that are part of your iris and another piece of tissue called the ciliary body. The virus can get into that epithelial tissue and replicate, and then it can spread from those epithelial cells into nerves that innervate those. So, for instance, your iris. I think most people know that if you look at a bright light, your iris constricts, and then if you are in the dark long enough, it dilates. And so that muscle action is due to innervation from your autonomic peripheral nervous system. So your sympathetic and your parasympathetic ganglia that innervate the eye. And because that wiring is coming in to control those muscles, the virus will see that and then enter and move into it, just like it would into a sensory neuron. And
when it enters these iris nerves, where would it end up?
So it's going to end up in two places. It's going to end up in the superior cervical ganglia, which is the one that we look for. And it's also going to end up in something called the ciliary ganglia, which we didn't look at in the study. And so those are sympathetic and parasympathetic circuits.
These are outside of the eye?
These are outside of the eye. The ciliary ganglia is just on the outside of the eye, and the superior cervical ganglia is further away. And so it's that longer distance transport to the superior cervical ganglia that we were examining in the animals.
I just want to jump in because, at least according to the pictures, it looks like you were in the aqueous humor, unlike what Alan was saying.
Oh, okay.
Oh, sorry. I probably misunderstood what he said. Yeah,
So Vincent said aqueous humor, and I said, "Do you mean vitreous humor?" But you actually mean, and then you described the actual injection site.
Correct.
Yes.
Sorry.
Okay.
Yes. So we're in the anterior chamber where the vitreous humor is the majority of the volume of the fluid behind the lens.
Right. That's the jelly that's actually in the eyeball.
That's in the main part of the eyeball.
Right.
That's right. And
the aqueous humor is just under the cornea.
That's right. And
so the vitreous is where they find Ebola in some of these people, right? Let's pause briefly here and acknowledge the presence of Dickson Despommier.
Hello, everybody.
How are you,
Hi,
Dickson?
Hello, Dickson.
I'm well, thank you.
You know, Dickson, I thought you were sitting here this whole time. I didn't know that you were
Surprise!
Well, we were just talking about injecting
I heard.
herpes viruses into the rat eye.
Oof.
Yes, that's right.
Because this is a good model for tracking, and it's bigger, as you say, so you can get more of everything.
And it's important to acknowledge that this is not a natural route that the virus would ever use to infect its host.
No.
But it does offer some nice advantages as a model.
Okay.
One, it's an immunoprivileged site, and so we're working with outbred animals, and it's nice that the differences in the immune response cannot cause variation in the results. So that's one of the main advantages.
And I just want to interject that this is leading to some of the coolest experiments in the paper, in my opinion. So.
Okay.
Yeah. So I guess what you did is to put the wild. Now, again, this is pseudorabies, right?
It's a pseudorabies, so it's very neuroinvasive and highly neurovirulent as well. So these animals are not going to survive this infection for a very long period of time.
All right. So I guess you put wild type and your K442 mutant virus in, right?
Yes.
Okay. Tell us the story.
Okay, so the story is if you put the wild type in there, and this has been known for a long time, that the virus will, in fact, enter those nerves that we were talking about. It'll travel within the axons as like a marathon runner, so they're going to be very processive and keep on going. They're going to make it to that to the cell body in the part of the peripheral nervous system called the superior cervical ganglion, and they will dock at nuclear pore complexes, inject their DNA, and now you have the virus has established an infection in the peripheral nerve.
Mm-hmm. And you can remove those peripheral nerves and find virus in it, right?
You can certainly do that, and so that's one of my collaborators' skills. They dissect out the superior cervical ganglia, and I should say for most of these experiments, we use a virus that has been a recombinant version of the virus that has been designed to encode a fluorescent protein, and so that just makes it very easy then to assess whether the neurons in the superior cervical ganglia have in fact been infected.
Okay.
By a simple fluorescence assay ex vivo. So that's the wild type. And now if you put the mutant into the system, what you see is that we get a phenotype that reciprocated a phenotype of our DUB mutant from our prior study. So what that phenotype was is that the virus is no longer making it to the nervous tissue. There's no evidence of infection. So that was the initial observation.
So can you assay infection in the epithelium where you first inject the virus?
You can. And so what we did in that case is we looked at the iris. So that should be the primary site of infection. Mm-hmm. And when we looked at the wild type as well as this lysine mutant and also the catalytic inactive DUB, all of these were replicating in the eye similarly.
All right.
Admittedly, the DUB and the lysine were a little bit reduced in their ability to spread throughout the iris, but it was not substantial.
Okay, but both the DUB and the K442 virus could not get out of the eye, basically, right?
They can't get out of they can't go down the nerve.
Right.
And for me that was a very exciting result because on the surface it sounds like okay, you have a mutant phenotype in two cases and that's okay, that's nice, but it's actually I think more interesting than that. The interesting thing is that the DUB mutant I would hypothesize at this stage that the DUB mutant would represent a virus that is going to be stuck in a hyperubiquitinated state because it can't take ubiquitin off the protein. Mm-hmm. Whereas the K442R mutant, so the lysine mutation, can't be ubiquitinated. So that should be a hypoubiquitinated virus.
Right.
And yet in either extreme states, the virus isn't getting to the nervous system.
Right.
And you could think that I think Hitaplu, when he was first making the discovery that herpes viruses have a DUB, the hypothesis back then was that cells might use ubiquitin as a way to target and try to degrade viral proteins as an innate protective response, and viruses may have evolved DUBs to counteract that. But that doesn't seem to be what's happening here at all. Right. It seems that the virus needs to be in both states for some reason, and it almost now seems like this is a regulatory cycle.
Of course, you have no idea what happens when, right? All you know is that you need to be able to put the ubiquitin on and take it off in order for the virus to travel along nerves, right?
That's right. Neither one can do it. And so then it gets to the experiment that I'm going to guess Kathy was referring to that she liked.
Yep.
And so the question then is why? And we knew from our prior study when we were just looking at the DUB catalytic mutant that if you put the virus into the brain directly, it will infect neurons and spread through neurons throughout the brain and cause a lethal encephalitis. So let me just recap that. So if you put the virus out at the periphery, it doesn't get into the nervous system. But if you put it straight into the nervous system, it's fine. So the way I would phrase that is that the virus is still neurotropic. It's just no longer neuroinvasive. And so so we basically repeated that experiment here, put viruses straight into the brain. Now, when you do this experiment, there's another important consideration, and that is when you're in the periphery, such as the eye, but really anywhere, the skin or anywhere else, the virus, before it can get into the nervous system, it must first replicate in other cell types that are receiving innervation, because neurons, for the most part, are not exposed to the outside world. The exceptions there are probably in the nose with the olfactory sensory neurons that are responsible for detecting odors. But other than that, they're going to be buried in other tissue. And so if the virus to get access to them, the virus first has to replicate in that other tissue and then transmit into the nervous system. If you put it straight in the brain, you don't have none of that's true anymore. The virus has direct access to neurons. And so if a virus is neurotropic, it should replicate in the brain just fine. And that, in fact, is the case. If you put the mutant straight into the brain, it replicated and spread throughout neurons in the brain, just like that DUB catalytic mutant did and just like the wild-type virus. So it doesn't seem like it has a neurotropic defect. And then the surprise came. And the surprise was that when you put it straight into the brain, you can look to see where it goes. And specifically in this case, our colleagues put the virus into a part of the brain that receives visual input from the retina of the eye. And the reason for doing that is that these viruses, since they spread within axons, they spread in a very circuit-specific way from neuron to synaptically linked neuron. And, I should just take an aside here and mention that a lot of neuroscientists have used these viruses actually to map out circuits of the nervous system, specifically because of this property. And so when it comes out of the brain and goes to wherever that part of the brain is receiving input from, in this case, it's going to the retina. And so the retina is nicely isolated from the brain, physically, and so now you can take out that tissue that's some distance away and look at it. And what we see is that with a wild-type virus, of course, it spread down the inputs coming from the retina and then infected the retina cells themselves, and you could see that very clearly. And if you looked at the DUB mutant, so the one that can't remove ubiquitin anymore, so it'd be in a hyperubiquitinated state, the virus looked just like wild type. But if you look at this particular mutant, the lysine mutant, it can't do it.
And this is retrograde or anterograde?
So we haven't actually used those terms, but this would be retrograde. And so retrograde meaning that it's spreading from an axon nerve ending or terminal to the cell body.
Right.
Yeah, that's right. And so for the first time, we had a separation of phenotype between the DUB catalytic mutant and the lysine mutant. And it's also noteworthy to point out here that lysine mutant phenotype that we just described in the animal is consistent with what we saw when we cultured primary sensory neurons and infected, and it was a drunk marathon runner. It couldn't go long distance retrograde effectively, and that's what we're seeing here. It's not doing it here either. But the DUB mutant doesn't suffer from that defect.
All right, so you need both DUB and ubiquitination to get into the CNS. But both viruses are defective at that, right? But to getting out, you just need the DUB activity, because the lysine mutant, the one that prevents ubiquitination, is okay at getting out, right? Is that correct, or did I get it wrong?
It's okay at getting out of, say it again, the
when you inject into the brain, it doesn't make it out. The DUB virus doesn't get out to the retina, right?
No, the K42.
It's the other way around.
Other way around, yes.
Yeah.
So you need K42 does not get out. Sorry.
It does not get out of in this model. It does not get out of the brain to the retina.
So you need
the in and out are going to get a little confusing here.
That's why I got a little tripped up too.
Yeah. So the in is you inject into the iris and it goes to a ganglia out of the eye, and both viruses can't do that, right?
They can't do that.
But if you put the virus in the brain and see if it gets into the retina, the K442 can't do that or can do it?
It cannot do that.
Cannot do it. So you need ubiquitination to do that. Yeah.
But in both cases, it's retrograde transport that moves the virus through the neuron, right?
That's correct.
So it's just the neurons are oriented in opposite directions in these two different experiments.
Okay. So we're not talking about different transport.
So
okay.
I think in and out are not good terms to use in this. We're just talking about the ability to move along a neuron. So the two things, as I conceptualize them, are the ability to get into a neuron and the ability to move along a neuron. Yeah. Is that a good...
Right, in the retrograde direction. That's exactly right. Yeah.
Right.
But we saw in your cell culture model that only one of them had a real problem getting to the cell body, right?
That's right. So when you dumb down the whole system and you use a reductionist model where it's just a primary sensory neuron in a plastic dish, then the viruses, the DUB mutant is able to go down that axon and get to the nucleus, whereas the lysine mutant has the drunk marathon runner phenotype, and so it doesn't do it very well.
I just want to interject my question here, and maybe you'll get to it later, but what does this portend for reactivation?
Well, it may not pretend anything for that because with reactivation, the virus is going to go through. Of course, it's going to. So the reactivation we hadn't really talked about, but you're now going to have viral gene expression, new particles assembled in the nucleus of the neuron. These then leave the nucleus, become fully assembled, and then transport in the other direction, which would be anterograde from the axon initial segment all the way down to the terminal. And so it turns out the biology there is different than what's going on in the early step when it's going retrograde to infect the neuron the first time. And what that means basically is that these mutations don't necessarily have to have any impact on that latter step. Mm-hmm. And we have data now that supports that completely.
So you have a latent model that you can test that with?
Well, so it's not so much latency here. So you can go ahead with PRV because it's so virulent. It doesn't actually go latent in anything in its natural host, and so it will immediately go into a productive replication, and then those new progeny will immediately be sent anterograde down back out, and then you can so. In culture in the dish, you're basically saying instead of looking at 10 minutes post-infection, you're looking at about 12 hours post-infection.
Have you studied anything in terms of factors that lead to go from the brain back out to the eye? Is there certain causes of that, or is it just stochastic and one of these drunken runners heads off in that direction?
So the brain to the eye is just a model here, so we could look at retrograde transport in the absence of any kind of epithelial barrier. That's what it really came down to. And so in a normal person, like in a normal infection for a person, you usually don't get a brain infection, although it does happen and it's very traumatic when it does. That's herpes simplex encephalitis, about 1 in 200,000 cases reported in the USA annually. It's a terrible disease, but fortunately fairly infrequent. And in that case, it's not so much a question of how does the virus get out of the brain, but how does it get into it in the first place. Right? I'm not sure if I'm addressing your question or not.
Yeah, no, actually I was wondering is more generally, and other factors that lead it to go in the opposite direction down the neuron back to because I'm wondering just sort of my background is an electrical engineer, so I'm looking at given a fact how can you apply it and maybe use it to control other phenomena. So I'm thinking other varieties of other members of the herpes family like zoster virus. I guess at some point it goes back. General belief, as I understand it, goes back down a neuron and cause an outbreak of shingles. And I'm wondering if you've seen anything here that is similar with that other virus.
So you're talking about I think you're talking about the reactivation.
Yes.
Okay.
And reactivation normally in a normal set of circumstances, that's happening in the peripheral nervous system. And so I guess I was a little tripped up when we were talking about the brain specifically. So we're using the brain here as a model, but in nature, it's going to be in the peripheral nervous system. That's true both for varicella zoster, chickenpox, and shingles, as well as herpes simplex virus type 1. And in that case, the spread after reactivation, you get new particle assembly, the spread is down the axon in the anterograde direction. Which I believe gets back to Kathy's question. So in that case, I guess the question is, what factors are necessary for the virus to do that? And that's something that we've been studying a little bit on. Normally I would defer, if he were here, to Lynn Enquist, because this is something that his lab has spent a lot of time on. But I'll just be a surrogate for a moment and say that there are some viral factors that seem to be important for this step. There's one in particular that's a membrane protein that the virus encodes. It's called US9. And it does seem like there are, in fact, viral factors are necessary to make that process efficient, but maybe not in all cases absolutely required.
So, Greg, can you now tell us, can you put together a model for deubiquitination and ubiquitination and these properties of the virus?
All right, so let's bring it together. So what we got then is a virus, a set of circumstances now where the virus has to be in a ubiquitinated state or a deubiquitinated state, and it seems like what we have is not so much a cycle where it's constantly going back and forth between the two states, but rather just two sequential events. So for the virus to first get access to the nervous system from the periphery. So if it's replicating, let's say, in this case, in our experimental design, it was in the iris of an eye, but I would imagine the same would hold true for the mucosa, sort of right around your lips if it's replicating there, that the virus first has to be able to not only replicate in that tissue, but then disseminate deeper to the nerve endings and gain access to those nerve endings. And you my thought prior to this body of research was that that's probably just a casual process. You get lots of virus replicating, lots of particles going everywhere. Neurons are going to get hit, and this virus is neurotropic. It's going to go in and do its thing. But it turns out this shows that that's a poor assumption. For the virus to get access to those nerve terminals, it needs something more. And what it needs in this case is the deubiquitinase activity. So the DUB is going to put it into the unubiquitinated state. The unubiquitinated state is what we call in this paper state 1, invasive state 1, that allows the virus to transmit from epithelia to nerve endings. And that is we say that because it matches all the phenotypes that we've described, and we can go through that again if we want to. But so then state 2, once it gets into the nerve ending, it now has to go into the ubiquitinated state. And that ubiquitinated state is necessary so that it can processively move down the axon nonstop, so that it is an effective marathon runner, and then get to that nucleus. And if it's not, and it's a drunk marathon runner, it can't do it anymore.
Now, to get from state 1 to state 2, it's going to have to turn off DUB?
Yeah, good question. So that's right. So the question basically is, why is it that when it gets into the neuron, it gets ubiquitinated if the DUB is still around? Shouldn't the deubiquitinase still remove the ubiquitin so that it can never go into that ubiquitinated state that it needs to be in? And absolutely, the DUB, we have to infer, I don't have any direct evidence of this yet, but we have to infer that the DUB is not able to do that once it's in the neuron. And so either the DUB is inactive, or it's being clipped off perhaps by a protease that's unique to the neuron, or there's some other kind of conformational state. But I would hypothesize at this point that there's something there that prevents that's unique to the neuron, and that's something we're very interested in. So
if something in neuronal cells that can regulate DUB, that's what you're looking for.
That can regulate the DUB.
Yeah. Right. It's likely to be a cellular protein, not a viral protein, right?
Or it could be more complex and involve elements of both.
Yeah, it could be another viral protein that responds to a cellular protein. On and on. You could go on and on and on.
Go on and on. Yeah.
Goes all the way down.
Turtles all the way down.
Yeah, ubiquitin all the way down.
Yes, that's right.
I used that joke on another podcast this week. We were talking about transcription factors, and he said, well, there's this transcription factor, and it's controlling another transcription factor, and I said, yeah, it's transcription factors all the way down. Yes. And he actually got it. Yeah, it's a turtle transcription factor, he said. Now, is this, so you and I talked about, in general, viruses that invade the CNS. This is likely to be a herpes virus specific switch, right?
At this point, that's how I'm thinking about it. Yes. And we should mention that, when we talk about CNS, infections, herpes viruses, like we just briefly mentioned, rarely do that.
Yeah.
That would be herpes simplex encephalitis in a person. So they've evolved to exploit the peripheral nervous system as their niche and do it in a very avirulent way for the most part, and it's in their native hosts. Whereas other viruses that cause CNS disease, those diseases are very rare for any of them. So Vincent, since it's polio in your case that you've been studying all your career, we can use that as an example where about 1 in 100 to 1 in 200 people that were infected during the 50s outbreaks in the States were getting poliomyelitis. Those are the estimates. For the most part, it's just an enteric pathogen,
right,
and of little significance in those circumstances. And so when it does get into the nervous system, because it's rare, the question then becomes, what is normally keeping it out?
Yep.
But with herpes viruses, when they're going to the nervous system, we don't actually know what the frequency is. If you do get exposed to that person with a cold sore, how likely is it that you're going to have a productive infection? I don't think that anybody has an answer to that question, but we know it's going to be very frequent. And because 80% of us have it in our nervous systems, I have to assume that's ability to get in the nervous system is far superior to other viruses that do it only on rare occasion.
I want to just quote the line that you have in the discussion about this. This feat is remarkable given that most unrelated viral infections occur one cell junction away from peripheral nerves, yet few viruses invade the nervous system. Yep. And they're just one cell away, and they don't do it.
Right.
It's really cool.
It's interesting, isn't it? I mean, on the one hand, you could say some viruses just aren't neurotropic because they don't use the right receptor that would be present on a neuron. But in many other cases, that's not the case at all. And so we could compare this to a number of other, classic neurotropic viruses that can cause, in some rare instances, very severe disease in humans, and why it is that they may be so different in this regard from herpes simplex virus and its related cousins.
So the basically the barrier. What are the barriers of the CNS to virus infections? Are they mostly physical, or are they physical and immunological?
Yeah, there's certainly both. So interferon is a big component here. And of course, we're talking about primary infections in people that have yet had an opportunity to develop an adaptive immune response. So under those unprimed circumstances, a lot of different things can happen. And, a lot of neuroinvasive viruses are arboviruses of one flavor or another. So flaviviruses are a big one. And then when these viruses get in, and the alpha viruses, when these viruses get in, they're being delivered by a mosquito, and they ultimately will develop a pretty severe viremia or a high-load viremia. And that gives them the opportunity potentially to get to the brain via the blood. Now, one of the barriers that you're asking about is the blood-brain barrier. There's also an equivalent barrier to this cerebral spinal fluid. So there's a blood CSF barrier, and there's also a blood-retina barrier. But basically, these barriers are restrictive enough that viruses normally just can't simply diffuse across them. And so they have to somehow be compromised if a virus is going to go one of those routes, and that certainly can happen. But that doesn't appear to be what the herpes viruses are doing. They don't usually cause viremia. They can under unusual circumstances, and they can in newborns who get exposed during the birthing process if the mother has an active infection. But for the most part, that doesn't happen. So they take a neural route, and so neural routes are diverse, and the herpes viruses are using these sensory neurons as their way in to the peripheral nervous system. But the other one that is very suspicious is the olfactory route, which a number of viruses can certainly use, at least in animal models. And there is some very interesting reason to suspect that when herpes gets into a person's brain and causes that terrible outcome, that it could actually be doing it through the nose.
Wow. That's what we used to think years ago for polio, and that was disproven.
Right. Now in polio, though, I think it's very clear that it can go up a motor neuron after. But so of course, as everybody probably knows, this is what Vincent did with some of these classic experiments, where if you take a mouse and you put polio, you inject it intramuscular, let's say into a hind limb of a mouse, the virus will then enter the nerve endings of the motor neurons that normally make that muscle contract, and then it will go retrograde up that motor axon fiber and get into the spinal cord. And replication in the spinal cord then gives you the flaccid paralysis, which is a classic presentation in humans of poliomyelitis. And so you can model that very effectively using mice. Of course, the mice have to be a special recombinant mouse that has the receptor. And so, a lot of people don't appreciate this, but you can turn the clock all the way back to the 1920s, and there are people doing experiments like this for many different viruses, including herpes viruses. And if you put herpes into the hind limb of a mouse, guess what happens?
Does it get in the CNS?
It goes right up the sciatic nerves, motor neurons, and gets into the lower spinal cord.
Interesting.
If you do the same thing with West Nile virus, you get the exact same result in animals. This is much more recent research. So I think basically if you put something with a needle into a muscle, if the virus is neurotropic, it's very likely that's going to be able to do this in an animal model.
So that's what we need to do with Zika.
Now, I bet you will go.
Yeah. You think it will go right in? Yeah.
I think it's more likely than not, because it seems like there's no reason at all to suspect that herpes viruses normally are going from muscle up across a neuromuscular junction and up a motor nerve to the spinal cord. That's not how the disease presents in humans.
Right. But it's not going to happen with every virus, right? It's only with certain viruses that are able to enter nerves.
They have to be neurotropic.
And they have to be able to travel up the axon and so forth, yeah.
Although the traveling up the axon bit may not be as complicated as one might think. I mean, most of these viruses are able to do this. I think that are neurotropic, and most all of them are using endosomes as a way to just basically get on a bus and write it downtown to the neuron cell body. Herpes viruses are different in that regard, that they fuse in, and so they don't actually go. They are not using an endosome to carry them. They have to actually have mechanisms to tie into that machinery directly to go retrograde. So I think most things can probably go in an endosome and go retrograde, especially depending on how you model it, because it's known that if you do any kind of damage to a neuromuscular junction, those motor neurons ramp up endosomal retrograde transport. And so basically you're opening up, not only by injecting polio into the muscle of an animal, you're not only just positioning it right at the front door of the nervous system, so it's ready to go in, but you're basically now putting a conveyor belt there so that they do go in, they get whooshed down right to downtown. And so I think we're kind of intentionally or unintentionally favoring this to happen in these animal model systems.
It's probably worth pointing out too that the barriers of getting in the CNS are pretty good, but once a virus gets in, then you have big trouble because the brain is not well protected.
That's right.
It has many of the defenses are damaging. Many of the innate and adaptive defenses would be damaging to the brain, so they're somewhat suppressed, and if a virus gets in, you have big trouble.
And you should also say the converse, that not all viruses that are able to get into the brain are going to be debilitating.
That's right.
So the classic is polyomavirus, JC virus, that apparently is in the brains of lots of people doing absolutely nothing as far as we can tell in terms of disease. Yeah. Unless you start treating with drugs to block lymphocytes from crossing the blood-brain barrier, and now the thing just goes kind of haywire. And now you get nasty infection.
Yeah, good point. Yeah, that's really good. So do you have any plans to do this with herpes simplex?
Yeah, we've already started.
Okay.
Yeah, so we've made all the mutant viruses, and so far everything is panning out just like you would expect. So it appears to be fully reproducible in all these regards.
Okay, so that's cool. That outcome will be cool, and then of course how ubiquitination, deubiquitination are actually controlling these movements is the other key, right?
Is the other key, yeah. And then I would say third, what exactly are these barriers that are intrinsic in us or in a mouse or a rat? What are these barriers that the virus is overcoming and using this what we've termed the ubiquitin switch? Right.
All right. That's great. Anybody else? Yep.
Yeah, I just wanted you to comment a little. I also like the part in the discussion where you talk about the evolutionary implications of this, that it would be hard to get both this, the need for ubiquitination and the need for deubiquitination, and that's maybe why herpes is so unique.
Right.
Yeah, so to just expound that a little, I think the fun thought project here is, and Vincent and I were talking about this on the phone before, that if you're an evolving animal and viruses are a problem getting into your nervous system because it's one of the worst places you could possibly have an infection, then you might think, okay, evolutionarily speaking, we're going to evolve to have a brick wall that's going to be built around the nervous system so viruses can't ever get in. And then, of course, the viruses evolving so much faster are going to figure out a way to get a sledgehammer and break through that wall. And so you're never going to win. Now, this is rampant speculation, but I think it's fun to talk about.
That's the Donald Trump approach.
Well, yeah, he built. Yeah, so right. So okay, so without getting
just dig under it.
Yeah, you could do that too. There's a lot of different ways through. So then, what is an answer? Is there some way that you can keep the viruses out? And of course, adaptive immunity is a great answer if you can wait for it to kick in. But if you need something quick, one way we're thinking about this is that you could set up a barrier. Or a pair of barriers that are going to have to be breached in succession, and the barriers are going to have to be designed so that they're exactly opposite one another, so they're polarizing. So imagine, the recent way I've been thinking about this recently is like light, is using a light analogy. So light, if you send it through a polarized filter, becomes polarized in that orientation. But if you then have another filter that's rotated 90 degrees. Mm-hmm. Behind it, then no light can get through both filters. Right. So if you think of the light beams as viruses, they can evolve to go through the horizontal polarizer or the vertical polarizer. They can have one or the other, but they can't ever get through both. The ubiquitin switch is now allowing a virus to be horizontally polarized, get through the first filter, then switch and go to vertical polarization and get through the second filter. And so now can robustly get past the barriers every single time. That's the kind of the grandiose model of how we're thinking about this. And I think another interesting thing that this could impinge upon a little bit gets back to polio, which is one of my other favorite viruses. And that is that with polio, you have this phenomenon of quasi-species. A lot of RNA viruses, of course, are quasi-species. And there's some nice work that came from Roland Dino's laboratory and also Carla Kierkegaard and Julie Pfeiffer that basically showed that the quasi-species phenomena, having this basically population of genetic variants of the virus present all at once as a swarm, that diversity needs to be present during the infection for the virus to effectively get into the nervous system. And what I would suggest is that maybe that is another solution to this problem, although not, I would say, is an elegant one. That basically, if you have a big complicated population, if one type of virus is, let's say, horizontally polarized, so it can get through that first filter, that first barrier. If it can bring along one of the other ones that's vertically polarized with it, if it can carry it along, then the other one's free to get through the next one. And so then when you look in the brain, all you're going to see is the vertically polarized virus, because the horizontal one won't get through the second barrier. And you take that and you put it in an animal and you say, hey, look, it's not neuroinvasive anymore. And I think that's how they were thinking about it. And I think this kind of model actually predicts that as well.
I was thinking of an internet security analogy, which would be two-factor authentication, where you log into the website on your browser and then you get a text message with an additional login, and herpes virus has uniquely figured out how to steal your phone and your browser.
Well, I'm in the process of writing a review, so I'll see if we can work that in.
That's a good one. I like that, yeah. You could work the FBI into it, too.
Yes.
Who's the FBI in this scenario?
Yeah. There's something going on right now between cell phones and FBI.
Well, no, I know that. I'm just trying to figure out how it works into the biology.
I don't know. That you can do it. You can work that out yourself. I'm just giving you stuff here.
Yeah, I'll call Tim Cook and see what he says.
We have actually a relevant Zika-related email, I think, that has to do with CNS invasion. Maybe we'll get to that. But anything else before we move on this topic?
First author is Nicholas Huffmaster.
That's right. So he was the wonderful graduate student who pulled this through, and it had a lot of stamina because, like many projects, there were some good days, but there were a lot of tough days in between. And so he is now actually on the job market, and because of personal reasons, he had to move to San Diego. And so if you know of any virologists in the San Diego area that would like somebody of this caliber, he is currently looking.
And the other authors are Sollers, Richards, Pickard, and, of course, Smith.
Yes, and Richards, Alexia,
just to fulfill the formal requirement that we list the authors.
and Alexia Richards is a postdoc in the laboratory who has some very interesting work that you'll be hearing about very shortly about how viruses get in the nervous system as well.
Cool.
You want to stick around, Greg, or do you need to go?
I can stick around if you'd like to have me.
Yeah, we'd love to have you.
Sure.
Absolutely. This episode's also sponsored by Microbe Magazine Podcast. This is a podcast developed by the American Society for Microbiology. It's a monthly show hosted by Jeff Fox, who is the current topics and features editor for that magazine. If you're a member of ASM, you get it every month in the mailbox. Each episode of this podcast corresponds with an article in the issue of Microbe Magazine. An example is episode 8, which focuses on producing yeast strains for making lager beers with new flavor notes. If a beer can have a note, I guess it can.
Sure.
You can find Microbe Magazine podcast on iTunes. Just search for it, or you can listen at microbeworld.org/MMP. And I got to say, in science, MMP is a matrix metalloprotease.
Yes.
It's just
and lager beers are good for what ails you.
All right. Now, this could have been an all Zika episode easily. All of the emails are about Zika, and there were a couple of papers out this week. Let's do a few of them. The first two, actually, that I put here were taken from the website microbe.tv/TWiV. And I'll just summarize. The first one is from Bodan, who tells us about the weather in his Amtrak train.
Going through North Carolina, apparently.
Your Zika podcasts have been terrific. I have never been so interested in mosquitoes as now. My interest in the past has been limited to metallic mosquitoes, syringes. Let's see. Okay, it was very helpful for Vincent to mention the continued problems with congenital rubella syndrome. And I had mentioned that there are 100,000 cases a year, and he is wondering where I got that number from, and he found a lower cases, like 33,000 for 2014. So I got it from the WHO website, which I linked to in my article. But the point is that's a lot, and it could be all prevented because we do have a vaccine. And he also gives us a listener pick, and he says, if you can distract me with fascinating scientific trivia, I can try to do the same to you. Sends a link to a TED Talk by Daniel Wolpert, and it's really cool. The real reason to have a brain is because it helps us to move. Okay, so that's pretty cool. You should check that out. Let me just paraphrase one more, and then you guys can read the others. The second one's from Kayla, and this 2 episodes ago we used her picture of Boromir and said one does not simply walk into the placenta. And she's very happy that we used it, and she's working on a Hannibal Lecter pop culture reference right now. That's going to be scary. She asks a question about Zika. If Zika is endemic in areas like Brazil, why is there a seemingly suddenly correlation? Has the connection always been there and we're just seeing it now, or has something changed? Well, the point is it's brand new to Brazil. It's not been there before. Serological surveys show that. And this is why it's spreading so crazily, because there's no population immunity. And so this connection between the recent new introduction of Zika and the rise in microcephaly has to be dealt with. Thanks to Alan for the continued meme fodder. All right, Kathy, can you take Deborah's, please?
Sure. Deborah writes, Dear TWiV team, I listened to the Zika discussion, TWiV 376, and there are times when the team was not sure about the physiology of pregnancy, the placenta, fetal blood-brain barrier, maternal immune system, etc. While you are all experts in your fields, I think it would enhance your discussion exponentially if you add an expert in maternal fetal medicine when discussing pregnancy. There would be less guessing and trying to remember from prior readings and education, and would lead to a much crisper and reliable discussion. Similarly, I would suggest adding a content expert occasionally when you are applying lab science to clinical work. Thank you for your generous sharing of expertise. Deborah, and she's an RN. Looks like
Jeremy was supposed to pick up the slack.
Right.
Yeah, really.
Right. But
some of the problem is that sometimes we start talking about things that we don't plan to talk about, right?
Right. We don't know what way we're going to go.
So it's hard, and then we can't just pull a subject matter expert. I don't have.
We did have a subject matter expert on placenta biology.
Yeah, she was there.
So
actually, Deborah, I would like to know exactly what we didn't know, or you just don't like the meandering, reaching it. Part of the whole thing is the journey, right?
The process of getting there, because I think what we're doing is, we're exploring these topics the way we would as a group of scientists talking about them, and I think that's what most of our listeners are into.
Yep.
Yeah, I like Vincent's description of this as a conversation that a couple scientists would have in your office or over lunch or something like that.
Yeah, it's not meant to be tight and planned, right?
Right.
It's meant to be a bit meandering. And I think that's what it's appealing to many people, right?
Right. But we can try and anticipate when we might need a subject expert a little bit more.
Yeah, we can. Yeah. I mean, I think Jeremy fits some bill, but he's certainly not a obstetrician.
No.
Right.
So anyway, I just moved one up, and I think Alan is next. Can you take Fernando, Alan?
Sure. Fernando writes, "Hi, TWiVirate. I was doing one of my backcountry skiing trips the first half of February, so I'm only now listening to episode 374, where you talked about norovirus infection in cruise ships. Now, is Fernando the ship's agent?"
No, he is not.
Okay.
No.
I spend quite a bit of time in backcountry mountain lodges, which are ideal noro habitat because people arrive with their microbes from all parts to share a small cramped space for a week or so. Lodge managers have painfully learned to be obsessive about containing the virus. All surfaces, door handles, railings, bathrooms are repeatedly disinfected with industrial-strength bleach. Staff serves all food and individually wrapped sandwiches, if at all possible. Some of the more remote lodges lack running water and standard bathrooms. Guests with suspicious symptoms are segregated to separate bedrooms and bathrooms. Instructions for bathroom and hand disinfection are prominently posted, and more. I don't have statistics, but the anecdata I've observed suggests that lodges have become much better at keeping noro at bay than they were 10 years ago.
Another reason not to go to backcountry.
You might see flowers, too.
Yeah, sign me up.
No cherry blossoms.
No angiosperms?
Backcountry and cruise ships, they're both checked off my list.
Well, he's talking specifically about cabins that are off in the woods that you ski to.
Yeah,
so
right,
the backcountry and packing your own tent and sleeping in the woods by yourself, you're not going to pick up anything that you didn't bring with you.
Frankly, I can't podcast from there, so I'm not going.
No, that's true.
The next
I like
one,
the word anecdata.
Yeah, anecdata is very good. Josh writes, "Hi, TWiV crew. Thought you might be interested in this article in Nature News about Dave O'Connor's, who was a guest on episode 260, Badgers Go Viral, work to make his lab's Zika data public in real time, similar to the recent commitment by many journals to make Zika papers open access, which you've already discussed." And there's a Nature article where he talks about he's doing experiments and he's releasing the data real time. You could go to this website, I think, and see the results every day.
There was way too much swearing in my lab notebook to do that.
Alan, do you want me to tell people about your lab notebook?
No, that's good.
Okay. Full disclosure, I work at LabKey Software and collaborate with the O'Connor Lab. We develop LabKey Server, the open source data management system the lab is using internally and to share their data with the public. I started listening to the podcast years ago and was happy and surprised to hear Dave's voice on that episode. Josh. All right, that's great. What Dave is doing is great there. I think Kathy's next, right?
Okay, is this Aileen?
Yes.
Is that how you pronounce it? Aileen. Dear TWiVzics, as a chatty big fan, here I am again. I try to keep silent, but it's bigger than me. I work as a regional coordinator of vector control here in Brazil, and I would like to bring some information about the use of larvicide. The use of larvicide, pyriproxyfen, is part of vector control, and she gives the Brazilian acronym, all over the country, not only in the Northeast. The same way with the adulticide. Now we are using diethyl succinate, which is malathion. Before 2014, we used Temifos as larvicide. Now, that's some brand name that I'm not sure what that is, but okay. The program includes periodically evaluating the safety and effectiveness of larvicides and adulticides to combat vectors. We follow the protocols. The numbers of applications and dilutions are planned and guided by program standards, and those are WHO guidelines. I work at the Secretariat Estudial de Saúde do Rio Grande do Sul, State Health Secretary, in the south of Brazil. Here we only have had one case of microcephaly associated with Zika virus. The mother had traveled to Pernambuco in the northeast of Brazil during the first trimester of pregnancy. Despite the low number of cases of microcephaly, Rio Grande do Sul was the only state that suspended the use of pyriproxyfen in potable water, a non sequitur, as a friend said. In fact, this is not a big deal because we have never used the larvicide in drinking water here. We continue working normally as we always have, applying larvicide at deposits that we cannot remove, but not in drinking water. There's a protocol to use it in drinking water, but we haven't adopted it. Our reality is quite different from the Northeast, where the population faces the difficulty in accessing drinking water, making it necessary to store it. We don't have this kind of problem here. As Alan has predicted, as a result of the suspension, people now are not very receptive to larvicide. Best regards, Eileen.
And Temafos is actually the generic name
The pyropry
of an organophosphate larvicide, and apparently they switched to in Brazil. There was a resistance problem with Temafos, and they switched to pyroproxyfen, which is a pyridine-based larvicide, and then this conspiracy theory came out, or what have you, and obviously now, in many cases, they're not using pyroproxyfen.
So there's a problem here that I detect because I have studied mosquitoes in the past, and I was part of a team that looked at hormones and how they controlled mating behavior, etc. So the dengue carrying mosquito, which is the Aedes aegypti mosquito, is a tree hole breeder. It breeds in temporary bodies of water. So spraying generically for the adults or putting a larvicide in drinking water doesn't address the problem of just cleaning up the environment. And that's the trick that Walter Reed, once they discovered that the yellow fever virus was transmitted by the yellow fever mosquito, which is the same mosquito, by the way, cleaned up the area around the Panama Canal for temporary breeding sites, and that solved their problem without the use of either insecticides or vaccines or any other behavior. So a cleaned up, routinely policed environment is the best hedge against spreading either dengue or yellow fever, or in this case, Zika.
Well, that's true, but it's the reason these larvicides are used is because in situations where, for whatever reason, that can't be implemented.
Understood.
So yes, it's absolutely the case that if you clean things up and you don't have containers of stagnant water sitting around, and everybody cleans out their gutters on time, then you don't have a mosquito problem. But getting that done is much harder than spraying. So
I wonder, is there, since you've studied mosquitoes, is there a precedent for mosquitoes moving in result to just ecological change? Because in Brazil, there's been a lot of replanting of crops, to produce alcohol for us, and constantly in the Amazon, there's for hundreds of years, just indigenous peoples, burn soil and grow. Yeah. Would something like that displace the mosquito, or is that not a factor?
It's a changing target, and indeed Brazil used to have a population of Anopheles gambiae, and so what the heck is that doing there? It came from the Gambia in Africa, and they spent a lot of time cleaning that mosquito out, and when they did, of course, the rates of malaria went down. But this one's different. This is an egg stage that survives droughts and can hang around for quite a while undetected. And then as soon as you've got a big flood, the next thing you know, you've got these clouds of mosquitoes. So I'm wondering why they didn't also sense an increase in, let's say, dengue, which is a very common infection that this transmits, in the same area that you had these epidemics of Zika. Same mosquito.
Well, the background level of dengue is pretty high there.
So it is, yeah.
But there are seasons for it.
So I think it's just a measurement error or changing standards?
Well, that's a good question, because if the same mosquito is transmitting both of these, and if the transmission of dengue is as high as Alan indicates it is, and I'm sure he's right, then why wasn't Zika higher than to begin with, too? Why is there such a fluctuation?
Well, because Zika's a more recent arrival.
Just arrived there, yeah.
They've had dengue there for many years.
So it's just going to be high. Both of these are just going to be high, is what you're suggesting.
And, right. Zika is just settling in, and so it's just getting going, and I think eventually it's going to be an area where you have endemic dengue and chikungunya and Zika virus
right.
transmission.
Right.
We got my friend Jorge Pontual, who's a journalist from Brazil, sent me a link to a new PLOS Neglected Tropical Diseases paper that was released yesterday, which is basically a short note in which a 20-year-old woman from Salvador, Brazil, had induced labor because of her fetus was detected as having severe microcephaly and hydrops fetalis. And so they induced labor, and the infant was born dead. And they did RT-PCR in various tissues, and they found Zika virus RNA in neuronal tissues, but not elsewhere. Mother reported no symptoms consistent with Zika infection. Now, this goes along with the case we talked about last time, where an aborted fetus was shown to have microcephaly and viral RNA. In fact, the entire genome in the brain, and there were also two. So that was the entire genome in the brain. Now, the interesting thing here, if you go over to ProMed Mail, there was a comment by Dr. Steve Seligman, who said, and I think this makes sense, it is possible that brain tissue in cases of microcephaly becomes susceptible to Zika virus infection by a mechanism such as diminution of the blood-brain barrier. So what he's saying is maybe the Zika isn't causing, but it gets into the brain because there is a problem, a physical problem with the baby. That's a tough one, but I think
which I think we actually mentioned a couple of TWiVs ago as a possibility.
So I wrote what's his name? I wrote Seligman, and I said, "You have any evidence?" He said, "Nah, like you, I'm a virus guy. I don't know anything." So okay. So that's that. And then there was also yesterday a case on another circumstantial case of Zika sexual transmission in Eurosurveillance, where an Italian man went to Thailand for 10 days. He came back to Italy, and a day later he got a rash. And this was over a year ago, so they thought it was dengue. And then 15, 20 days later, his girlfriend got a rash, and they figured they were both dengue. And now they just pulled the serum samples out because they said, hey, let's look for Zika. And they both are seropositive for Zika. So the article says it's most likely sexual transmission, but I'm still sticking to the idea that how do we know this? Because it could have been close personal contact that transmitted the virus, right? We don't know that it's sexually transmitted. And yes, they did admit to having sex, but I don't think you could say that's the way it was transmitted, and I don't think they should have it in the headline.
Right.
It doesn't sell papers, though.
It does sell papers.
Kathy has just posted a good link.
Right. So back on Dickson's question, it rang a bell with me that I thought that there had been evidence of an increase in dengue. So I found this article from May 13th, 2015. The Brazilian Ministry of Health reported 746,000 registered cases of dengue fever and 229 confirmed deaths since January 2015, which is a 234% increase in cases and a 45% increase in deaths from the previous year.
Look at that.
So maybe it's a mosquito increase.
Indeed.
And they're both as a consequence. Yeah,
so what would cause an increase in mosquitoes in a population?
Weather and land use.
Yeah, that's right. That's right. And migration. That's a very high migration area. These people live in these areas like Piauí, which is a Brazilian district, and they migrate to the mine regions in the interior to earn a living, and then they return back to their homes after the season for mining is over, when the rainy season hits. So there's a lot of movement afoot also, and then you've got these climate change issues to deal with as well. So this is really going to be a difficult issue to address, because it's just vector control that's going to win the day in here, I think.
So if I can interject, so with this question of is there something that predisposes the unborn child to the virus, it sounds like the discussion has been, what is it about that unborn child that has something wrong with it? Maybe the blood-brain barrier is compromised or whatnot. Yeah. But is there any reason not to equally suspect that there's something about the pregnant mother herself, that she has a barrier deficiency for protecting her own blood supply to the baby?
Right.
Well, that's a good question, and we do not have a placenta expert here.
Right.
So,
nor are we willing to speculate.
I do not wish to, but we could ask
to irk the ire of Nurse Ratched.
I could send I could send Carolyn Coyne that question, because it seems reasonable to me.
Yeah, it seems like an unborn child is going to have a lot of susceptibilities, regardless to me, from everything I understand.
I mean, the question is whether such barrier deficiencies are known or not. I just don't know.
Right.
Have there been any studies to see if there's any correlations in socioeconomic status, or if this is broad-based?
Well, the Zika transmission is going to occur predominantly in poorer areas, because that's who's exposed to the mosquitoes.
Well, that's where the debris is the highest too, right?
Right. That's why they're exposed to the mosquitoes.
Exactly.
So So I'm not sure that you would detect anything over the background or apart from the background of that.
The same doesn't apply to Anopheles mosquitoes, by the way, many of which does require a clean water environment in order to breed. So it's quite a different situation in terms of that.
We have 3 email left. Should we finish them?
Yeah.
Sure.
All right. Who's next? I forgot. We got Mitchell next. Is it Kathy?
Sure, I'll read it. Dear TWiVsphere, recently I have seen many articles discussing the sexual transmission of Zika virus through semen. The first case suggesting sexual transmission was in 2008, and we since witnessed over a dozen cases, albeit not confirmed, of sexual transmission of Zika. In my immunology course, I was taught that the testes were an immune-privileged site, similar to the blood-brain barrier and the placenta. Yet Zika particles have been found in semen up to 62 days post-infection. My question is, if Zika can be found in the semen of infected men, that would suggest it crosses the testis barrier. And if this is the case, could a similar mechanism be used by the virus to cross the placenta as well? I understand these cases are not confirmed, and this might be wild speculation on my part, but can immune-privileged reproductive sites play a role in the spread of Zika virus via sexual transmission?
I would just correct Mitchell in saying that we didn't find particles in semen. We found RT-PCR fragments
nucleic
of the nucleic acid, right? I think that's reason. Yeah, I mean the testes are immune privileged, and
so Ebola virus finds its way.
Ebola is there. Fragments of Ebola genomes are there.
Sure.
The eye as well.
Yeah.
And couldn't they be spread? Yeah, it could be, but we just don't know yet. I don't know about the placenta. He says could a similar mechanism be used to cross the placenta?
They're very different structures.
They're very different, so I'm not sure you could say it would be similar.
How about in nursing mothers? In mother's milk, and then getting across that barrier, because that's another barrier.
Sure. We talked about a paper on flu where it was implicating milk as a transmission mechanism, remember? In the mammary gland?
Right.
Now, we have since witnessed over a dozen cases. I don't think that's quite right. I mean, I like to say when there's a journal article, then it's a case.
Right.
So we have had, we had the 2008 Colorado case. We have Zika in semen. And then we have, there was a report, and there are 2 other reports, including this new one that I mentioned today. So maybe 4 suspected cases, but...
Right. But then there's the New York Times headline from February 24th, 14 new reports of Zika's sexual transmission in the United States.
Yeah, and I bet they're not confirmed. They're just...
Oh, of course not.
Yeah. But, they put it in the headline so that...
Yeah.
All right.
Sex sells.
Sex does sell. All right, the second is from Patricia TWiVites. I am very intrigued by the discussion of Zika, primarily due to my interest in human biocultural evolution, being a trained biological anthropologist. My own personal interest is in hominin evolution. However, teaching a basic introductory course that satisfies a natural science lab requirement at the university necessitates me talking about human infectious diseases. I typically use sickle cell malaria and lactose intolerance as examples in the class, but the last few years with the Ebola virus, the nurse that traveled to Akron caused quite a stir, and now Zika has allowed me to use proximate examples in classes. Thanks for keeping a relatively informed non-virologist up to date. I began listening to TWiV during Ebola virus and have not stopped. Nothing like a good outbreak to get
That's right.
our listeners. My question is primarily with Zika transmission across the placenta. I understand through my own studies and teaching basic embryology at medical schools that inductions or hiccups during development can result in massive fetal abnormalities. While it is unclear when Zika might infect a fetus, I agree that it seems likely an early developmental infection might be more likely than late, given neuronal embryology. So could it be possible that moms become infected with Zika at a non-clinical symptom level prior to getting pregnant? Because Zika is in their system, the placenta and blood supply to developing fetus is produced when Zika is circulating in the mom, so that virus does not necessarily cross a developed placenta boundary, but is present in the onset of placental development. Just curious, this is not my primary area of interest. Current weather in northeast Ohio is rain, wind, and temperatures 40 F, 4C, and also beginning of the mud season, particularly bad considering my daughter rides horses regularly. Mud season's also starting in Massachusetts.
Yes, it is, yes.
Patricia is a lecturer at the University of Akron.
Akron.
Akron?
Akron.
And like Oregon?
Yeah, Oregon, Akron.
Tire capital of the world.
Yeah, it's close to where I grew up.
Tire capital, really?
Yeah, they make a lot of tires.
And and mosquitoes, right?
Well,
well, they have tires.
No, those go for they go to Japan for retreads, and they come back, and then they've got the mosquitoes inside.
All right, so I don't know how long the virus would stick around, right? It depends on the timing, and we don't really know how long it persists. We're going to learn this at some point. So your scenario seems to me reasonable depending on how long the virus is present, right?
Yeah.
Right.
Yeah.
Last one, Alan, is from Lisa.
Okay, so Lisa writes, "Greetings, TWiV team. My name is Lisa, and I'm a graduate student in micro and immunology at Vanderbilt. I've greatly enjoyed listening to your last few episodes regarding Zika virus and its potential link to microcephaly. It spurred an incredible conversation I had this past weekend with my grandmother, who was a nurse in obstetrics from the late 50s to the early 90s. During her tenure, she saw a number of anencephalic and microcephalic newborns, though she thought the increase in microcephaly, especially in Brazil, reminded her more of babies she saw born to mothers who had taken thalidomide. This is mostly because there weren't any clear causes in most of the instances of microcephaly, whereas thalidomide was at first correlatively and then causally linked to birth defects. Side note, I've recently learned that thalidomide is in use again, though now as a cancer drug with very careful screening to ensure it isn't given to pregnant women. She had a great question that I don't think has been directly addressed in the last few episodes, though if it has and I missed it, I apologize. Has there been an increase in miscarriages in Zika-affected countries? And if so, are the brains and skulls of the miscarried fetuses appropriately sized for their gestational age? She thought perhaps if the infection happened early enough, the mother may miscarry instead of taking the baby to term. I'm a longtime listener to TWiV, TWIM, and TWIP, and I was curious if you ever thought about making TWi this week in immunology. I'm training in a bacteriology lab, and listening to your various podcasts has greatly expanded my knowledge of all facets of microbiology. I just wish I had known about them before I took my qualifying exam 2 years ago. It's currently 47 Fahrenheit, 8.3 Celsius, as I walk to lab with a high in Nashville today of 62. Thanks for all you do. And then she follows up again. Hello again, TWiV team. I had emailed earlier in the week about a possible correlation between Zika virus and miscarriages, and I just wanted to follow up with a paper that was published today. Zika virus infection and stillbirths, a case of hydrops fetalis, hydroencephaly, and fetal demise. And this is from the... Was this the one you just linked?
Yeah.
Okay.
Yeah.
Yeah, it seems as though there may indeed be a link between Zika and fetal demise, yes.
So I don't know how... I mean, her question about whether miscarriages have gone up, I don't think we know. That's a single case report, right?
Yeah, and we talked about that with Carolyn, and she said it's really hard to quantitate the number of miscarriages. We don't even have a good handle on how many there are, period.
Good.
You know, and that, you know, with certain of them, you're not going to have any evidence of the fetal size or embryo size.
Yeah.
So, yeah.
So today I emailed our friend in Brazil, Esper Callis, and he's on the ground there and dealing with this. And so he's going to come on in a couple of weeks and actually talk to us.
Oh, exciting.
Good.
So we can ask him these questions.
Perfect.
As for immunology, as you know, Audiomunity has a podcast of their own. But yes, I am working on an immunology podcast. I'm trying to get together the right crowd. And it may come out in the next year or so. We'll see.
You could call it Twins this week in non-self.
I could definitely
Brought to you by Bill and Tom, or
Twins.
B and T for short.
Wow, that's good. Let's do some picks. And let's see. Maybe we'll start with, hey, Greg, are you still there?
I'm listening.
Do you have a pick?
Do I have a pick?
You don't even know what a pick is, do you?
Do you have a shovel?
I know
That's
exactly.
right. Pick
I know
or
what
shovel.
a pick is. I wasn't prepared to offer a pick.
Okay.
Come back to me at the end.
All right, I'll come back to the end. Let's go to our guest, Mark. Do you have a pick for us, Mark?
Yes, I do. It's a book by an author, a researcher named Gregory Burns down at Emory, and it's called How Dogs Love Us. And it's an interesting study. He does research with functional MRI on reward behavior drug issues. And one day was wondering, is his dog more than just is it in any way sentient, or is it just very well trained? So we decided, maybe I could take it into the lab and do an fMRI and find out. Well, the first thing was he had to train the dog to lie still. So he describes how he built a wooden,
easy in an MRI.
a wooden chamber in his garage about the size of an MRI, and tried to train the dog to feel comfortable into that, and then get him into the MRI itself and not get scared and start to move around when all the magnets and noise occurs. And long story short, he finds that when you present with food or when you present with the owner or someone else in the family, you see the similar activation patterns in the dog's brain that you see in the human when they see food or they see a loved one or a family member.
That's cool. You have a dog, Mark?
Yes, we have 3.
Wow. You're dog lovers, right?
Yes, we are.
In fact, your Twitter photo is a dog, right?
Yes, my dearly departed but great Cocker Spaniel.
Oh.
Dickson, do you have a pick?
I do. It was an interview between CNN and Bill Nye the Science Guy. It was on about a couple of days ago. And indeed, this is probably the reason why we're not hearing from Vincent about his appearance on that show, because Bill Nye was not available that day. And here's why. He imagines himself insinuated into the audience of the GOP debate with all the candidates in front of him, and then he begins to question them with regards to their views about climate change. And the questions continue to get more pointed until finally he's asking them whether or not their science background exceeds that of the IPCC experts who constructed this scenario to begin with from real data. And at that point, I presume that all the candidates will cave because none of them have science backgrounds. So why are they so critical of climate change, or why don't they adopt it? Because it's a form of political terrorism to actually purposefully reject the science that might predict the next 100 years of hardship for an entire country. How dare a presidential candidate not talk about this issue in public is beyond anyone. And Bill Nye brings us out in spades.
No one presses them. There's no tough questioning.
Well, there was tough fighting the other night, but that was like little kids.
And if there is tough questioning, they're not going to respond the way Bill Nye hopes they would.
No.
They would not cave. These characters.
You think they're too good for that, Balin?
They would retrench. They would just dig in deeper, and they know their audience. Their audience is not Bill Nye.
Yeah, of course.
Well, so Bill Nye got to say his piece anyway. So
sure,
deal
yes.
with it.
So this is just published today, and I think he did this interview yesterday. So I was asked on Tuesday to appear with Bill Nye on the Star Talk radio show, which is a video and an audio podcast. So I was going to go down to the studio, and they wanted to talk about viruses. And then Wednesday they canceled it, and this is why.
Okay.
So I don't get to talk to Bill, but maybe they'll ask me back at some point in the future.
They ought to. He could use the TWiV bump.
Yeah, you're right.
And you can tell him that you saw him in the elevator.
That's right.
I would tell him that.
That TWiV bump was caused by a mosquito bite, by the way.
Alan, what do you have?
I have just an article that came out in the Open Notebook. Don't know how many people in the listenership read this, but this got a fair amount of attention among science journalists. So these folks, the writers here are Cynthia Graper and Catherine Gammon, but there was a whole volunteer group behind them who did the actual work of the counting in addition to them. They went and they counted bylines on science news stories from a bunch of high-profile publications, The Atlantic, The New Yorker, New York Times, etc. And they were interested in finding what the gender distribution was of science writers at these publications, which was an interesting thing to look at. And they found women are pretty well represented in general among science journalists, which did not surprise me at all. But the interesting finding here is that men are disproportionately overrepresented as the authors of long feature articles, and women are disproportionately overrepresented as the writers of shorter news articles. And anybody who's in the news business knows that the long articles are the ones that give you the prestige and the bigger paycheck. Mm-hmm. So this ties into a TWiV arc that we've had about gender skews in the sciences, and this apparently carries over to some extent, although it's a different world, but it's it's interesting to see this show up in the context of something like science writing.
So you have to go below the initial numbers, basically.
The initial numbers look like a pretty good, some publications skew a little bit one way or the other, but very small differences. But then when you actually dissect that into the types of articles being written, you find that the high profile assignments are disproportionately going to men at most publications.
Huh. Did they correct for, tenure as a writer and other factors?
I don't think they drilled too deeply into that, although I can tell you from experience that the turnover rate in science writing is pretty high, so you don't have to worry too much about that. And that it has been a fairly balanced or even female-dominated field for quite some time. So there are certainly plenty of female science writers who have decades of experience.
I didn't know about this site. This looks great.
It's a neat site, yes.
A lot of cool articles here. How Rebecca Skloot built the immortal life. Stuff from Carl Zimmer. Oh, cool. I'll have to mark
it. So it's sort of the backstory on science journalism.
Nice. So you read this?
Periodically, yes.
Nice. That's very nice. Kathy, what do you have?
Well, last week I mentioned the News and Views article by Anise Lowen, which didn't end up getting into the show notes. That was in the same Nature issue as the paper itself. But you mentioned the article that was, Dickson mentioned the title of the article that was by Andy Melle.
Right. And, that's the one I had in the show notes, yeah.
Right. The avian influenza virus polymerase brings ANP32A home to roost. I remember Dickson saying that. Right. But this is all a segue into the fact that because I'm reading the things in hard copy when it's a journal I have like Nature, I read Anise Lowen's News and Views. But next to it, I couldn't even finish her News and Views because I was so attracted by the story that came next, which is a story about sources of Chaco wood. And this is Chaco wood in Chaco Canyon, these big homes that were built by Native Americans. And they've been able to figure out where these great halls or great buildings, whatever they're called, where the trees for them came from by looking at basically the rings of the trees, and they can it's called tree ring sourcing. It's not dating, and it's just a really cool article that I wouldn't have seen otherwise. So,
this is neat.
Yeah.
So
where did they come from?
Yeah. That is I'm sorry. Go ahead.
No, that's all.
That is one reason to keep reading the paper journal, because you wouldn't find it if you're just drilling down to articles.
It's like the same thing with the demise of bookstores. You pick up something just because it's next to something that you were looking for.
Yeah. All right, I have 2 picks. I've been fascinated by this FBI Apple debate about opening up an iPhone, and I found a wonderful blog by a forensic scientist. So this is a science pick, right? And his name is Ziarski. I forgot his first name. Let's look at about. He is a forensic scientist who specializes in iOS, Jonathan Ziarski. And he is just writing beautiful posts about this whole thing, and they really get in deeply into the issues. And I find it fascinating, the protection built into the iPhone and what you need to do to get around it and what the FBI wants and so forth. So if you're interested in that, you should check that out. And then yesterday, of course, Apple's motion to vacate the FBI order was published. And this is a somewhat long document written by lawyers, but in fact is remarkably readable. And I, as I read it, I just think of how much it cost to write it. But for example, it starts off, "This is not a case about one isolated iPhone," and it's very good. And they make some really compelling points and talk about the precedents and so forth, what's happened before, and what laws we have on the books to deal with this. So I'm fascinated by this whole thing, and these are 2 good sources to check out more if you're interested.
Neat.
And that now, Greg.
So I have a question.
Yes, sir.
About that issue. Why can't Apple decode this phone? And if there's nothing incriminating in the messages on the phone, just tell the FBI we didn't find any incriminating evidence.
Because it's not that simple. Okay.
No.
It's very complicated, and we need about an hour if you want to know the answer.
Basically, though, the FBI wants them to break barrier function on the phone, and so all sorts of viruses are going to get it.
And then there are dozens of people who are waiting to get their phones cracked for similar reasons.
I know that.
And once there's a precedent, and to crack it
will spread.
It will get out
everywhere.
You mean
in China,
viral?
Every government will want it. Yeah.
Just look at you think, oh, I'm going to give this to the FBI and it'll be safe. But Snowden took all the secrets from NSA, and so nothing is safe anywhere at Apple or the FBI or here
what's being asked here is not, could you please unlock this one particular iPhone? What's being asked here is, could you open a door that would allow mass surveillance?
Yeah.
Uh-huh.
Or even more than that, it's would you go out and build a door opener?
Would you? That's the other thing being requested is, would you please install a back door that would make it easy for us to do these sorts of things that we have a track record of doing? Yes.
And the thing is that they picked this case because it's an emotional trigger.
It's very high
right?
profile. Yes.
But in fact, this phone probably has nothing on it because he had two other phones that he destroyed, and but they know that public sentiment may drive this anyway. So read about it. I've been reading a lot. It's a lot of fun. So Greg, do you have a pick?
So I'm going to mention two things, and one of them is 2 weeks old, so you probably already did it and I missed it. But I just want to say that the announcement, I think it was 2 weeks ago, of the discovery of the gravitational waves, I think is a nearly once in a lifetime kind of discovery, and that's just watching the scientists talk about their discoveries publicly for the first time. I just couldn't help just thinking they're. I mean, that's the Nobel Prize right there. Right? It's got to be. I mean, it was just so amazing.
Greg, did you see the take on that on the Colbert Report?
I didn't.
Or Colbert Tonight Show. Check it out. Brian Greene was on night before last, and he had a mini version of the detector.
Oh.
And he showed how it worked. It's absolutely fascinating stuff.
Oh, great. I'll take a look for that.
Yeah,
and the other thing I'll just briefly mention is there's this video that recently got posted online from Boston Dynamics, and they have their new version, their Atlas robot.
Right.
You can just Google search for this video because you have to watch this thing. I mean, all I can tell you is that humanity is coming to an end because it's terrible.
You got it.
Wonderful.
We all agree.
All right. Thank you, Greg.
Sure.
Sorry to put you on the spot, but
that's right. You gave me a second to compose.
Part of the TWiV experience, and that will do it for TWiV 378, and we have managed to hit the 2-hour mark. Once again,
bing, bing, bing.
You can
ring the cowbell.
Yeah, I should.
Got it right over here.
You can find TWiV at iTunes and also at microbe.tv/TWiV. And you can send your questions and comments to TWiV at microbe.tv. Our two guests today, Greg Smith from Northwestern University School of Medicine. Thank you, Greg.
Thank you. This was a pleasure.
We loved hearing that work. And when you get the next big aha moment, we'll have you back.
All right. Look forward to that.
Thanks so much. And our in-studio guest, Mark Fuccio. Thank you so much, Mark, for stopping by.
Thanks for your hospitality. It's great to be here.
Did you find it long and interminable?
No. But the difference, compared to listening on a podcast, is you can't hit the rewind button and re-listen to something.
Just wait till Sunday. You can do it all again.
You can do it again Sunday. So do you often do that?
I do. I'm driving, especially sometimes especially if sometimes Rich Condit, he tends to speak rapidly. So you need to go back and re-listen and reparse some of his words.
I see. So you don't do ours at 1.5x, right?
No, I couldn't imagine anyone doing that.
All right. Kathy Spindler is at the University of Michigan in Ann Arbor. Thank you, Kathy.
Thanks. This is a lot of fun, and I learned a lot.
Alan Dove is at turbidplaque.com. You can also find him on Twitter. Thank you, Alan.
Thank you. It's always a pleasure.
Dickson Despommier is here at Columbia University, where he is an emeritus professor. You can find him at verticalfarm.com, the father of the vertical farm. Thank you, Dickson.
You're welcome, Vincent.
Sounding like the grandfather of the vertical farm.
Yeah, maybe you are the grandfather.
I couldn't resist. I just couldn't resist.
Of the vertical farm. Did you have a good class today?
I had a great class today. It was terrific, in fact. We had a great time.
This is my 5th podcast this week.
Wow.
That's incredible.
Did a lot of podcasts, but I'm still ready. We could do another one. Anyone want to do another?
Sure. Come on.
Do too.
You know, like Ernie Banks for the Chicago Cubs, his favorite saying was, "Let's play two." You know, he always used to just jump out on the field. He was so enthusiastic.
That's good. We need enthusiasm.
Let's play two.
We are here at TWiV. We are enthusiastic, if anything.
We're placenta poor in knowledge, but enthusiastic nonetheless.
I'm Vincent Racaniello. You can find me at virology.ws. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.